Okay, so today's lab is relatively short because we'll talk about throat culture. So before that, I would love to spend the time talk about aerobic respiration, the calculator of the ATP. And we will repeat it again in the lecture section because today we'll talk about TCA cycle. But the whole picture I would love to draw on the blackboard. So you can follow me, take notes, because this will be in the lecture exam. And I want everybody to get it, because it is important, and you learn in different classes. Although we do the recording for the lecture, but it's possible somebody never to go there or never look at the video even. So in the lab section, we want to talk about that. Okay, so the overall thing is aerobic respiration, and the accumulated, the ATP calculation, which means how many ATP going to be generated. Okay, so let's go from the beginning to the end. I'm just go ahead and just do the structure. We will have the first item is glucose. So glucose, 6 carbon. This is a Fischer configuration we mentioned about a couple of times. So first one, glucose. Then glucose will become glucose 6-phosphates. And that is 1, 2, 3, 4, 5, and 6. So happened on the 6 carbon. So the second one, you can just draw exactly the same. Basically no difference at all. H, OH, here, OH, H, goes here, OH, H. So difference is right here. We have CH2O phosphates right here. So we can draw like this, okay? This will be glucose 6-phosphates. Then we're gonna go here. We'll do an isomerization. So the isomerization will be changed the configuration like this. So we have CH2OH. This is OH. This is I always say this is gonna be the key. Okay, H, OH. This is sometimes up and down. It doesn't matter because it's a L and D configuration difference. So this is I won't be picky in the exam which direction you go. CH2O, this will be our phosphates. So what is this? This is fructose 6-phosphate. Okay? Then the fructose 6-phosphates, we're going to draw a little bit, go up right here, will become still the fructose, but we're going to add a phosphates there. So add the phosphates in the carbon 1. Go here with H, OH, here, OH, H. Go down here will be H, CH2O. I use a phosphates is add a P there. So this one we call it fructose 1,6-biphosphate. So at this moment, you have to be careful about these phosphates. Where they comes from, it honestly, very obviously coming from ATP, really storage the original. So ATP become ADP and phosphate right here. And this is isomerization, so nothing happened. When fructose 6-phosphates become fructose 1,6-biphosphates, where they add this one here, this is obviously here happened another, which is ATP hydrolyzed become ADP and release phosphate. Okay, so this phosphate is actually added right there. So at this stage, we call it an investment. This is the whole thing we call it an investment and it is a 6-carbon stage. We were using is 2 ATPs. So we write minus 2 ATPs. Okay, then we move on to a branch. A branch, you should know very clearly, it looks like this. Okay. Now, of course, the beginning is glucose, and the end is pyruvate. So what we are finished, right, is around here. We finished. Okay, 6 carbon we talk about. Then we're going to talk about these 3 carbon. Okay, so the 3 carbon, I'm going to split there, so I'm going to draw right here. Okay, 3 carbon, and I said in the lecture, it is very obviously, this is symmetric configuration you can break down in the middle. Then break down in the middle becomes 3 carbon. So what it is? CHO aldehyde. We have CHOH and CH2OP. So what is this? Carbon number 1, carbon number 2, carbon number 3. So glycerodehyde 3-phosphate. Okay, then we're going to go down here. We're going to add some phosphates. So what we're going to become is COO. This P will be there. And then go down, become CHOH, CH2OP. You have 2 phosphates. So 1,3-phosphoglycerate. Okay, so then let's go down here. When you go here, when you have 1,3-biphosphoglycerate, then something have to be losing. Okay, so you're gonna become you're gonna become COOH, CHOH, become H, and CH2OP. Three phosphoglycerate. Then the next step is easy, which means this phosphate you move to the top. Is that right? So what are going to become is COOH, CH goes here, you become O, and the P is in a 2-carbon position. Then you go down here, you have CH, because it's moved up, so we become CH2OH. So that's what we become is 2-phosphoglycerate. Okay, then at this stage, what's going to happen? We basically going to lose water later on. Okay, later on we're going to lose water. So we're going to go here, a little bit on the top. So we'll become COOH. So you go here will be CO. So same thing, P. And you go here, this double carbon becomes single, and here is CH2. So what is that? This is a phosphorenopyruvate. Okay, phosphorenopyruvate. Then the last step, it goes down here. This phosphate is gone, so you become COOH, C2, CH3. This has become a pyruvate. Since we'll talk about the ATP generation, so we want to mention several things. When you see here, where the phosphates comes from, this phosphates comes from inclusion, okay? Then here, which is NAD, will be reduced, become NADH2. And when 3-biphosphoglycerate becomes 3-phosphoglycerate, this is one phosphate is gone. What happened is ADP using these phosphates becomes ATP. So there's ATP happens. Now 3-phosphate become 2-phosphate glycerate. It just to switch the position. That's called a mutase. Remember the enzyme called a mutase. But last here, which is enolization happened. This is process is enolase. So go here and the phosphoenolpyruvate become pyruvate. There's a phosphate is missing. Is that right? Where they go? That is ADP with phosphates becomes ATP. So there is a several things is important. Is right here and right here. These are 2 ATPs, which is 3 substrate level phosphorylation. Okay? But do not forget, once it is aerobic respiration, this guy here will be going through electron transport chain and generate ATP. And I will do the calculation, it is only 3 ATPs. Okay, because some of your textbooks say 2.5 because we're considering about the ratio of the phosphate and the oxygen and the phosphates, but we don't talk about here, so that's will be 3 ATPs. So how many? We'll add 3 ATPs right here. Remember, this is a branch. So this whole thing is happened twice. Therefore, we multiply by 2. How many generate? It is -2 plus 10, so equals 8 ATPs. Okay? Now we have this pyruvate comes out. This pyruvate we are going through a cycle. So hope we have a space here. So I have to draw a little bit small. First of all, there is a transition step. We'll just write here easy. So pyruvate, we're going through here, which is D-carbon dioxide, using SCOA. Of course, happened, which is NAD reduced to NADH2. So this is the one, which is COOH, CH, and SCOA. This we have the name called acidyl-CoA. Okay? Then the acidyl-CoA, we're going through a cycle. This is very important and everybody knows called citric acid cycle. There's the other name we'll talk about in the lecture. Okay? So here we just briefly. So this happened with acidyl-CoA will be there. So acidyl-CoA, I'll just write acidyl-CoA, which is COOH, CH, and SCOA. Now, by the way, this is a long name, okay? So we're gonna go through the cycle. First of all, very important, when you know this, you need to know what is citric acid. Citric acid is like this. COH, COOH, CH2, and COOH. This is citric acid. Okay, or we say citrate sometimes. Different textbook have a different one. Citric acid for bacteria, E. coli, it's make it a little bit easy. The next one, we will be directly become isocitric acids. In you can use, you say it's alpha cis candidate. We don't have this one here, so we make it a little bit easy. Okay, we don't have that one here. So COOH. What is go H go here, then OH go here, then we go H, then we go COOH. Okay, this is isocitric acids. Now, isocitric acids, we're going to go a chemical here, which is you're going to lose CO2, happened NAD reduced to NADH2. What is that? So that's chemical is COOH, CH2, CH2, CO, and COOH. This is alpha ketoglutarate. Okay, right here. Then the alpha ketoglutarate, right here, it's a little bit flat. It depends on how you write it. So COOH, CH2, CH2, COO, then you have SCOA. This is succinyl-CoA. Okay. The same thing when you hear happened is also losing CO2 and NAD reduced to NADH2. Now every time when these things happen, I would suggest you just circle it. Then you know that's important. Now succinyl-CoA will go a little bit flat, goes here, then becomes COOH, CH2, CH2, and COOH. This will be succinate. And remember here, there is something happened, which is a substrate level phosphorylation, is GDP using phosphates becomes GTP. Little bit different, not ATP. Okay, but that's substrate level phosphorylation. So we go up. When you see here, these are not really stable. Okay, so what happened is COOH becomes CH, double bonds, and COOH. What is this one called? This is called fumarate. Fumarate. Okay. Now where is this H goes? H goes with FAD, fulving adenine dinucleotides, becomes FADH2. This is fumarate. So we also circle it. Then fumarate go here. When you see these double bonds, it's very easy. You're gonna have to make them back. Okay, so we become COOH. This has to be up a little bit. This is not much. COOH goes here will be CHOH, CH2, and then go COOH. This is malate. Now remember, you have a malate gases using the beer, so this is malate. And this is usually add water, so nothing happens. And then you go here, the last stage, almost done, goes here. These you're gonna lose the water, gonna go again. Okay, you see here. So what happened? You see COOH, CO, CH2, and COOH. What is this? Oxaloacetate. Then you go back to acyl-CoA. Now right here, it happens again, which is NAD reduced to NADH2. So the whole procedure, we cycle them, we call it a citric acid cycle. How many ATP really generates? Here we have a 3 ATP. Here we have a 3 ATP. Here we have a 1 GTP, but it's ATP. We have 2 ATPs right here. And then we have 3 ATPs right here. And also do not forget the transition step at the top, there's also 3 ATPs. Okay, so how many right here? 1, 2, 3, 4. So 3 ATPs multiplied by 4 equals 12 ATPs. This comes out is NAD reduced to NADH2. Okay, now then you have 2 ATP right here. These 2 ATPs, which comes FAD reduced to FADH2. And do not forget there's a 1 ATP that is actually the GTP, which is substrate level phosphorylation. So you add up them together, how many? It is 15 ATPs. And then you have a 2 cycle. Because remember, this comes from here. This 1 cycle is right here, another cycle is right here. So 2 cycle, you multiply them by 2 is 30 ATPs. So for a bacterial cell, you add 30 ATPs cell here equals 38 ATPs. Now, what you learned in your class is biochemistry class. You calculate 36 ATPs. Where they comes from? Very easy to say is we minus 2 ATPs equals 36 ATPs, because that 2 ATPs is from to passing through mitochondrial gate. You can say a gate. Like you pay the ticket to go to see a theater or go to see a movie. Okay, so that's why it's 36 ATPs for eukaryotes. Okay, so that's basically the procedure where we talk about that and the calculation for everything. And I said the calculation everybody should require to know, and the chemical and the name will be the extra points. So for example, if you know glucose, you'll get 0.5 points. You know the structure, you'll get 1 point. So I will do is that extra points is 1.5 for each, then divided by how many you get it. Okay? Now the key thing is that here you need to write in. You definitely need to know the structure of the glucose. When you go here you write, you definitely need to know the structure of citric acids. If you don't know those two, it makes it very hard to finish the work. And I believe if you're taking biochemistry class as a senior student, you already know about these things, so it's a good for you to refresh. But as a junior or freshman, no freshman here basically, sophomore, it's a good time to learn. Now, but just try your best. Don't be too much pressure. It's not a biochemistry class. At least you know some. Is that right? At least you know the name. If you forgot about the chemicals a little bit, if you don't have enough time, that's okay. This is the opportunity for you to catch up something you're missing in the exam 1 and the exam 2. So let you be fully prepared to give you some extra. So we have another section next week like this. We'll talk about amino acids, the side chain, because we'll talk about the translation. That is also give you some extra opportunity to make up the points. Okay? So that is basically is a plan. Okay, so this is the first part. I will be just tear off right here.